It's with great pleasure that I again introduced Dr. Kiho Cho, who has over the years educated us tremendously on the different ways that the body responds to injury, to stress, to its own genome. So thank you again, and we welcome your thoughts today.
Thank you, Dr. Palmer. Almost all scientific hypotheses are based on some type of assumptions. For instance, whole genome is sequenced for humans and other species. And ribosomal RNA sequence can be used for classification, even for identification of different microbes. For instance, microbiome studies. And also, we were told that mice and humans share the same ancestor, and we evolved through random mutations. And all these assumptions are not that scrutinized that often. We scrutinize our hypothesis, whether the quality is good or whether we can test that hypothesis. But we do not scrutinize whether the assumptions, which are the foundations of those hypotheses, whether those assumptions are usable or the quality of those assumptions are well controlled. We do not scrutinize them that often. So as a part of my study about the assumptions, I like to discuss two key assumptions in biological sciences. That is classification and evolution. That is one. And another assumption is about the expression, detection, function, and interaction of a conventional gene. So the study subject, meaning humans, animals, and including plants, they are really complex and dynamically programmed. Therefore, the logics and systems underlying the normal and disease biology of these humans, animals, and plants are not that easy to decode. However, researchers introduce rather simplified and often clonal study designs, including relevant analytical tools in current biological research. So the question is, are the current assumptions, which are the foundation of biological studies, sound enough to produce meaningful data? Meaningful meaning that can we use that data for the care of our patient? Can we use the data to understand how plants grow? So this is a snapshot from National Human Genome Research Institute. They are saying that human, mouse, and other mammals share a common ancestor approximately 80 million years ago. So evolution can be defined this way. So formation of pleiotrophic life forms from a single origin of species through accumulation of random mutations during natural selection. So we were told that by NIH that mouse and humans share the same ancestor, and we evolved through random mutation. And is it really the case? In 1977, Dr. Carl Woese started using ribosomal RNA sequence for phylogenetic and evolutionary studies. So basically, he used the ribosomal RNA sequence to classify different microbes. But nowadays, scientists are using ribosomal RNA sequences to classify all different species. And one scientist said that it is as if Dr. Woese lifted a whole submerged continent out of the ocean. That is the new domain that Dr. Carl Woese introduced. That is archaea domain. So before the archaea domain, all living creatures were classified into two, prokaryote and eukaryote. So the life forms belong to prokaryotes was divided into bacteria and archaea based on this ribosomal RNA sequence. If we only focus on ribosomal RNA sequence or the conventional genes, which occupy about 1 to 2% of mouse and human genomes, then that makes sense that we evolved from the single origin. And also, mouse and humans share about 85% of conventional gene sequences. Sounds like we share the same ancestor. When we are looking at the human genome, about 1 to 2% are represented by conventional gene sequences. The rest are packed with repetitive element. Some scientists estimate maybe 90% of our genome is packed with repetitive element. The others are saying 66%. And some of these repetitive elements can move around, so they are called transposable repetitive elements. I like to believe that human genome system is a 4 to the n system. That means if we have 3 billion nucleotides, we are the system of 4 to the 3 billions. It is a number that, that is high to be called infinity. However, when we are looking at the human population on Earth, almost all 7.5 billion people share the same frame. Therefore, every single nucleotide and its position matters, not just the ribosomal RNA sequence. So Dr. Carl Wood used ribosomal RNA sequence to classify different microbes. And now there's a Bible of the microbial classification out there. But we decided to look at whether we can introduce high-resolution classification system using repetitive element profiles in different microbes or from the entire genome sequence. So we developed a system called genome signature image. Basically, we used CMYK scheme. We assigned 4 different nucleotides into the printer's CMYK. And then we picked the top frequency repetitive element from different microbes. And we developed a computation system to measure distance. And we test the system with the E. coli. We found that it is working. This is one of the interesting findings from that repetitive element-based GSI system. So extremely halophilic archaeons, they are supposed to belong to archaea domain, not bacteria domain. But as we did genome signature image, we found that 2 of them have different colors, meaning that they have completely different genomic profiles, even though they share a similar ribosomal RNA sequence. But when we are looking at the entire genome based on their repetitive element profile, they were so different from the others. Same with 2 strains from Prochlorococcus marinus. 2 are quite different, even though they share ribosomal RNA sequences. So we checked the entire sequence, and then we confirmed that those 2, which were supposed to belong to archaea, now we confirmed that they should belong to bacteria. So based on ribosomal RNA sequence, it belongs to archaea. However, if you look at the entire genome, we can reclassify them into bacteria. Basically, it's a change of domain. In evolutionary science, I assume that that is a big change because we are changing the domain name. Similar to the archaea to bacteria, now those 2 Prochlorococcus, which were supposed to be Prochlorococcus genus, based on ribosomal RNA sequence. Now, by looking at the entire genomic sequence, now we can confirm that they have to belong to Synechococcus. So we need to assign new genus name for these 2 microbes. We did not survey all the microbes. However, if we look at the entire population of the microbes out there that we studied, then we might be able to find more of these. So now we can question that whether the ribosomal RNA sequence information is sufficient for computing evolutionary distance or lineage. Maybe not. I show this, the fractals of the genome sequences a few years ago. So basically, we surveyed mouse and human reference genomes. We found that there are fractals in the genome. So the initial question that we had was whether our genome is ordered in terms of its structure, or is it just distribution of ACGT to make the conventional genes. So we tested whether our genome is ordered or not using simple dot matrix analysis. We did self-alignment, and we were able to find unique structure in our genome. We named it repair-T element array. That's RE array. We like to say that RE array is highly ordered, and RE arrays are unique for each species. So I'll just show you one RE array from each species that we surveyed. This is one example of human, mouse, dog, cat, rice, and poplar tree. We were not able to analyze dogs, cats, rice, and poplar tree in depth. That's because even though they claimed that they did whole genome sequencing, they did not do those non-gene areas that well, so we couldn't find many. But mouse and human sequences, they are relatively well done, so we were able to do an in-depth study. Just based on mouse and human RE array surveys, we found that they do not share RE arrays, even though they share 85% of conventional gene sequences. These RE arrays are highly organized and structured. So here's the question. If mice and human started from the same ancestor, if there are many random mutations for over many years, can we form this unique structure for each species? Random doesn't go with order, but mouse and human have unique ordered genome. So therefore, it is unlikely that mouse and human share the same ancestor, because random mutation cannot generate this species-specific order. So again, if I go back to the ribosomal RNA sequence-based classification and evolution, then when ribosomal RNA sequence cannot provide precise classification or cannot explain how the evolution between mouse and human can occur, then we can say that whether the ribosomal RNA sequence maybe is not sufficient for meaningful computation of distance and lineage among different species. So I'm not trying to say that the ribosomal RNA sequence-based system is not right, but I'm trying to say that we need to open up a discussion to see whether the ribosomal RNA-based the classification, even the identification of microbes are sufficient to find the meaningful data, so that we can use those data that we generate from those studies for the medicine and some other studies. So now, the single origin of species, that means dogs, humans, mice came from the same origin through random mutation. Then if that was from random mutation, then we can say that there's origin and random mutations. There is no specific pattern like an RE array in the genome of each of these species. Then when we see these, the unique RE array structures for each species, that means this random doesn't go with these the highly ordered structures specific for each species. Then we can challenge whether there is a single origin of species or not. Then we can propose this. Maybe dog has its own origin. There is a unique pattern from the ancestor dog. I tried to put D there, if you can see it. And then many years, random mutations, then now there's a German Shepherd, Chihuahua, Poodle, Retriever. So I like to propose that there might be origin of dog, origin of humans, origin of mice as well. So there are multiple origins of species and evolution through random mutations. So there are multiple origins, random mutations within each species. Now we have the population of dogs and population of humans and population of mice. And each of the genomes of these different species have unique ordered structure. Those ordered structure cannot be generated by randomness. So there are multiple origin of species, and if you look at the entire genome configuration, it makes sense. But if you are only looking at ribosomal RNA sequence, then single origin of species makes more sense. So tree of life or garden of life. Now I'd like to move to the next subject, that is the expression, detection, and function of a single conventional gene. As a graduate student and postdoc and earlier career in my science, I did not question all the information that I learn and that I read. So there's a gene, there's messenger RNA, and there's a protein. And each protein has unique function, and it interacts with other protein or proteins. So each gene may have unique function or maybe two unique functions. So when there's this, one gene or one conventional gene, so how many messenger RNA or protein isoforms are produced from a single gene? How many different functions do they have? And how many different combinational interactions with other proteins can happen with a single gene? And also, can we differentially identify multiple isoforms of a single gene with one or two antibodies? And do we really know how many different isoforms of a single gene is produced in vivo, meaning that in humans in this diverse population, not just in cell lines, in humans, humans with all different conditions, young, old, traumatic injury, infection, and others. So how many different types of isoforms of messenger RNAs or proteins can we expect in vivo? So with all these questions, we don't have the answer, but we have some evidence which suggests that there are a lot more messenger RNA and protein isoforms from a single gene. Therefore, it may complicate the detection and understanding of the function of those conventional genes. So this might be the current understanding. There's a conventional gene. There might be one or two different isoforms. There's a messenger RNA, proteins produced, a couple of different functions of a conventional gene. From our studies of our human glucocorticoid receptor, we found that human glucocorticoid receptor can make many different messenger RNAs and many different proteins. So each messenger RNA isoform of a conventional gene is formed by a combination of different exons. So if there are more exons, that means you expect more messenger RNA isoforms produced by a single gene. So far, about 15 exons are reported by the scientists outside of our lab. 15 exons of glucocorticoid receptor. However, last few years, we found there are 14 new exons in glucocorticoid receptor gene. So from a point of a single conventional gene, so now we are looking at 29 exons from a single gene. And again, each messenger RNA isoform is formed by a combination of different exons. So we really don't know how many different messenger RNAs will be formed from this combination of 29 exons. One of the uniqueness of our finding is that we did not look at any cell lines. We only look at the volunteers, and also we only look at the samples from burn patients from UCD Medical Center and Shriners Hospital. Maybe that was the reason why we were able to find more exons, because we are looking at the real human population, not just the cell lines. So if you ask me how many messenger RNA isoforms will be found from glucocorticoid receptor gene, I can answer that likely many, but I don't know how many. So we expect that we'll find more exons from this. So from this study, now, instead of having one or two messenger RNA isoforms, I expect that from a conventional gene, we'll find some more. In addition to that, we tested the function of those unique isoforms from these human glucocorticoid receptor gene. And that single gene can have many different functions. All these bars indicate the differential activity of those genes. So if we are looking at a single gene from now on, we can imagine that there are many messenger RNAs formed, and also there are many different proteins. That means there will be many different functions from a single gene. So instead of one or two functions of a single gene, we expect that there are many different functions of a single gene. So now it comes to the detection of this protein. So when we detect the protein, we use antibody. So let's say we have this antibody which can detect very well documented the full-length protein. It is bound nicely, so it can detect this protein. This cannot detect this isoform of that gene. And this antibody can detect this isoform of that gene. However, this protein is slightly different from the original intended target. But since it will share the epitope, it will detect. And these two cannot be detected with antibody. So even though you cannot detect any with your intended antibody, there might be the expression of the isoform of the target gene that you are looking for. At the same time, you might be detecting some protein that you are not intending to detect. So with a couple of isoforms produced, there's a limited number. This is our current understanding. Then our antibody will detect these two proteins. Even for these two proteins, their function might be different. However, when we are looking at multiple isoforms, then we expect many isoforms, and with the antibody, it might be able to detect some of these isoforms, but all of these proteins will have different function. But on your assay with the antibody, immunohistochemistry, flow cytometry, and others, you cannot differentiate whether you are detecting one species of protein or multiple species of protein from a single conventional gene. So now it goes to that high resolution or precision medicine or personalized medicine here. If we cannot differentiate these different isoforms from a single gene, we may not be able to come to accurate data. So let's say we're using antibody to identify specific cell types. This is a very common method for many different types of biomedical studies. So this antibody targets the protein of this gene. There's one conventional gene and antibody marker. So this is our goal. However, I can illustrate these different scenarios. So this antibody can bind to second isoform of the same gene. And this antibody binds to the target protein. However, the target protein is a minor population. The other isoforms produce a lot more on this cell. And this antibody, again, binds to this another isoform. Therefore, all these three different types which show the binding of the antibody may not represent the cells that you are looking for. So it can quite mislead your immunohistochemistry data and flow cytometry data, because your antibody may bind to second isoform or third isoform of the same gene. Or your antibody is binding really a minor population of the proteins produced by the specific gene that you are studying. This is another example that I can use to explain potential misidentification of cell type. Let's say this antibody binds to this protein to type those cells as a skin cells. However, this antibody binds to this protein, but this is an isoform of this, slightly different, so therefore different function. and there's a different protein expressed in this cell. So can we say that this is a skin cell? I hope not. So by using second antibody now, this cell can be conformed as a neuron instead of skin. I'm just using kind of extreme illustration and example here. So by not looking at the right isoform of a conventional gene, we might be misled. But with the current system, there's no way that we can find out whether we are misled or not. Similar here. So when we are doing immunohistochemistry, we'll use our antibody A, and all these antibodies are bound to these proteins. But as you can see here, there are two different isoforms. Our intention, let's say, was this solid protein here. Then initially, we can count it as a 25 positive cells. However, in reality, there might be 15 cells of cell type 1 with this solid protein and 10 of this isoform of the protein. Even though those two came from the same gene, but they can express completely different protein sharing the same epitope. Similar with the flow cytometry, with antibody A, we might be detecting 25 antibody A positive cells, but in reality, there might be two cell types. One is with solid protein and with the isoform of the solid protein. So there will be 15 of this cell type 1, there will be 10 of this cell type 2. So again, the same gene, two different proteins, and two different cell types. There's no way that we can tell with the current protocol. And I'd like to extend this further to the current gene manipulation or editing technology. If we are trying to manipulate the genes in mice and some other system, I'm using the word manipulation, meaning that kind of disable here. So the CRISPR is clustered regularly interspaced short palindromic repeat, which is very popular nowadays for the gene manipulation and editing. So let's say you edited the second exon. Then this editing of second exon will affect, let's say, the current limited isoforms. Then two messenger RNAs will be affected, and it will disable the function perfectly fine. However, if you decide to pick exon number 4, and if you did editing when there are two messenger RNAs, you might be able to disable the first isoform, but your second isoform is still intact. However, if you look at the multiple isoforms from a single gene, now it gets a lot more complicated. Let's say there are more than 10 different messenger RNA isoforms, and if we do manipulation on the second exon, then these messenger RNAs will be affected. But there are some other isoforms which are intact. But if you look at, if you did editing or manipulation on another exon, then we expect that there are more intact isoforms which are not affected by the manipulation. So when the CRISPR manipulation or editing is chosen for the study, they need to look at very carefully which exon should be edited, because depending on which exon you are editing, you may expect many other isoforms to be intact without any effect from the editing. And also, the CRISPR can be used for the correction of a conventional gene. Let's say there's a single functional protein, then correction should work fine. If there is a single messenger RNA, correction should be fine. You corrected this exon number 4, then now function is corrected. However, if you are assuming that there are multiple isoforms of a single gene, that means there are many isoforms. Then if you look at, if you are correcting exon number 4, there will be many corrected isoforms. Then you need to, when you are designing the experiment, you need to question which isoforms am I going to correct? Is that isoform number 1 or isoform number 8? So before we design any CRISPR-based editing, we need to look at the multiple isoforms of a single gene and pick the target carefully. So currently, the interaction between two genes are explained by mostly 1 or 2 proteins. So basically, there is a gene A and gene B, protein A, protein B interactions. Mostly based on this rather simple concept, the signal transduction pathways are established. A lot of them are established. So this is one of the signaling pathway. It's called the Wnt signaling pathway. So this is basically based on the simple interaction of one gene, two genes, protein A, protein B, and interact, without considering multiple isoforms from a single gene. However, if you assume that there are multiple isoforms produced by a single gene, then the interaction between gene A and gene B might be rather complicated. We need to evaluate all these combinations. Which combination do they use when they are belonging to Wnt signaling pathway? That will be rather complicated, but I believe that is worthwhile to figure that out before we establish any signaling pathway. So when we draw a signaling pathway now, we need to question which isoforms of the individual conventional genes participate in a signal transduction pathway. Is that the isoform number 2 of gene A, or is that isoform number 10 of gene B interacting in a specific signaling pathway? So when we are looking at limited isoforms of a single gene compared to multiple isoforms of a single gene, if you are looking at limited isoforms, we use the formula of fx equals to ax. I'm just trying to make it rather simple here. However, when we are looking at the multiple isoforms of a single gene, then we are looking at rather complex formula to understand and also even design our studies. But by introducing these multiple isoforms of a single gene, then now we can accomplish reproducible and accurate research. Currently, NIH stresses so much about reproducibility of the data. I never heard anything about accuracy of the data. However, I like to make it important that reproducibility is important. But if the reproducibility doesn't go with the accuracy, how good is it? So if you only focus 1 or 2 isoforms of a single gene, we might be able to reproduce that data over and over if you are careful enough. However, you will reproduce that data over and over almost the outside of the target. Then it's not meaningful. However, if you look at multiple isoforms of a single gene, and if you design and interpret your result, we might be able to accomplish accuracy of our data. And it goes with the personalized medicine, and also it goes with the precision medicine. Without this accuracy, reproducibility doesn't have any meaning at all. So our current biological assumptions sound enough to support meaningful hypothesis studies and outcomes. That's a big question. Do our assumptions support our studies and interpretations? I'm going to ask the audience, by looking at this illustration, from what city? Oh my God. Okay. And what is the name of this tower here? No. Salesforce, yes. This is the Millennium Tower. So we assume that our foundation is strong. However, if the Millennium Tower does not have strong foundation, it's not just a problem of the Millennium Tower. It's a problem of Salesforce and other big buildings around, because Salesforce Tower is nearby. So it is critical to evaluate our assumptions at least once in a while, instead of just go ahead with the assumptions out there. One good example is whole genome for humans and other species are completely sequenced. That's what they say, but that's not the case. So Dr. Wedwin Hubble, is our Milky Way galaxy the whole universe? Or are there other galaxies out there? I believe that there are other galaxies out there. Thank you.
Well, as I have said many times, you have to hear Dr. Cho's presentation multiple times to really understand what you're trying to communicate. But this time, I really found myself thinking that it's a very scary thought, and you present a very plausible argument for the fact that the assumptions that we've made to ask a lot of these questions about fundamental biologic properties, may be wrong. We may be reproducible and not accurate. I thought that was a great description. So why do you think we get it right as often as we do? When you just think about, the medicines we use to treat people. I'd be interested in your thoughts. I have thoughts of my own, but why does it seem that we do cure cancer a reasonable amount of the time? Or if there's so much variability in human response, why is our success reasonably good? Or maybe that's the assumption. Maybe it isn't actually very good. We just get lucky. What do you think?
So I see the biology or biomedical science as a game of probability.
Mm-hmm.
So some of those successes that you mentioned, maybe we are lucky to have that successful probability. So when we are looking at, for instance,
the right variation in response matching with this particular therapy,
right.
You get some people get lucky, some people are less lucky.
So let's say some drug is developed based on RAS signaling pathway. And there might be many different combinations of those genes involved in RAS pathway, but we are lucky enough to get the right isoform of individual genes. Then if we develop any drug based on RAS signaling pathway, then we might be able to develop the drug for some population.
Dr. Goodnight.
Dr. Cho, always stimulating. My question is similar to Dr. Farmer's and probably more naive. As you were talking, you talk about these multiple isoforms from a single gene, which for my simple mind is a scary amount of diversity and so forth. Would you take a stab at how this might be controlled? If you've got 15 different isoforms of the same gene, what allows that to proceed in an orderly function?
So when we are looking at just for the glucocorticoid receptor, we have patient number 32. He's about 50 years old. He was hospitalized for over 8 months with 9% total body surface area burn. We collect the blood samples at multiple time points during hospitalization and after discharge. We look at the isoforms of glucocorticoid receptor from this single patient. Depending on the patient condition, the stress conditions, we see different isoforms produced. So these isoforms, multiple isoforms of a single gene might be produced by a person's the stress conditions of their body. So I like to believe that as we get older, isoforms of a gene might be changing. So that, by only having about 30,000 conventional genes, it's not easy to explain our complex phenotypes, like the time-dependent and space-dependent phenotypes. So it makes sense that single gene can produce many different isoforms so that which can control the phenotypes of a person. And that the profile of those multiple isoforms will be quite different among different people. Even within a person, it will be changing over time and space. So for an accurate science, we need to account for those dynamic and complex changes in isoforms of the individual genes.
Let's see, Dr. McGuigan, then.
So if all mice share an orderly RE pattern, and all humans share a different orderly RE pattern, your hypothesis is that they didn't come from the same ancestor. But then on the other hand, they have 85% of similar genes in their chromosomal organized genes. So by probability, both seem like extremely unlikely scenarios that we're going to have separate ancestors but develop 85% of common genes, and also that we're going to have common ancestors but then develop completely different patterns of RE for all that massive amount of genomic material. Could it be that all that organization of the genomic material is regulated by just a few genes that then mutate it?
So to answer the last part of your question, so that means we need to assume that over 98% of the human genome should be junk. But that's not the case, though. So, and to go back to your 85% and the RNA data. So I'm neutral with any religion. So I'm not biased to any religion. I respect everybody's God. But if there is a creator, I'm using the word creator, I don't know. But if I'm a creator, now I'm going to make a dog, I'm going to make a mouse, I'm going to make a human. I have the basic ingredient for 2 eyes, 1 nose, 2 hands, maybe 4 paws. Anyway, I have those basic ingredients to make different species. Then why not use the basic ingredient to make 2 eyes instead of developing whole new? But now I'm going to make human as human. That means I need to introduce something unique for humans. So those are the junk DNA or RNAs, unique patterns, which can make human as a human, mouse as a mouse. So that can be my answer to your question.
It is interesting that there are not very many 4-eyed organisms that I can think of. Good point.
Thank you.
Dr. Cantor, you were.
Kiho, thank you, Mom. I'm drawn to the similarities with what quantum mechanics is to physical chemistry, right? Which is this notion that in the Bohr model, you know where the electrons are, but now we know it's a cloud, and you can only predict the probability of something being somewhere because once you observe it, it's already gone. Is the dynamics of what we're talking about here that quick, and should we abandon trying to look at like physical genes and physical proteins and so forth and try to predict the probabilities that certain things are happening? Like, is there a Schrodinger equation for biology and for genomics?
So before we abandon our efforts to understand the nature, I would like to propose that we, as researchers and scientists, should sit down and have a discussion. So what are we looking at? What level of complexity are we looking at? What level of dynamics? So to go to your complexity and dynamics, so I mentioned the human genome as a 4 to the nth system. 3 billion nucleotides, now we are looking at 4 to the 3 billionth level of complexity. Out of 4 to the 3 billions, only 7.5 billion combinations are selected to make humans on Earth. That means each position, each nucleotide is important. There's no junk in our DNA. However, if we introduce the dynamic nature of the genome, now it gets a lot more complicated. 4 to the 3 billions is changing over time and space, and it is unique for a person. Then that level of complexity is so high. So because of that, as you suggested, we should sit down and discuss how much we can do. There are certain things that we may not even touch, but we need to decide how far can we go. So I have an electronic engineer colleague. I've been working with that electronic engineer for over 11 years now. They confessed that in electronic engineering, they see the boundaries. They know how far they can go. So they push the limit because they know how far they can go. But in biology, I'm not sure whether we know that boundary because it's so complex. But a lot of times, scientists or researchers assume that they know quite some, but I don't think so.
It's the assumptions get us. Okay, Dr. Cantor, I think you're going to give him the last.
More of a comment than a question. It's an interesting talk. I just feel compelled to point out that you mischaracterized Darwin a little bit. The process of mutations may be somewhat random. I'm actually rereading the book right now. It's a great book for those of you who haven't read it. The process of mutations may be somewhat random, but what is passed on through natural selection is actually very ordered, because the only things that actually confer a survival advantage are passed on. So even though there is some randomness, it's really actually very ordered in terms of the process by which things are passed on through generations. And so that's an important point, because if you assume it's all random, it leads to a lot of the ability to say that there's no way that evolution could have occurred if it was totally random and you have these, just totally bizarre, adaptations and so forth.
So I agree with that a order in that random mutation, but I stress that order of those RNAs, those structures are so complex and highly ordered. So I mentioned that level of order. Yeah, I agree with you. Random mutation should have some order so that they can find a direction.
Well, thank you again for this incredibly thought-provoking presentation, and we'll take it back to everything that we do. Thank you.
Thank you.